All right, today's lesson is topic 7, which is environmental monitoring, and it is our last topic for this unit. So reminder, the unit test is coming up. All right, so environmental monitoring, this is also called ecosystem monitoring. It's a way to check the condition of an ecosystem by comparing the results of investigations that have been done at different times. So this is done because it helps scientists understand the impacts of disturbances and changes in the environment to try and reduce or reverse them. So both abiotic and biotic parts of an ecosystem are monitored. So abiotic would be things like temperature, soil quality, maybe the pH of the soil or the pH of the water. Biotic parts of a system would be any of the living organisms that are part of that ecosystem. So there's 4 types of monitoring. There's physical monitoring, and this uses satellites to track changes in landscape over time. So physical monitoring can show the changes to land that occur due to things like construction of cities, deforestation, anything like that. It's done using satellites. Environmental monitoring, this is going to track changes in climate, temperature, and weather patterns. Chemical monitoring, this is going to assess the quality of the air, the soil, and the water. So again, things like looking at the pH and the quality. And then biological monitoring, this tracks the changes in organisms or populations of organisms. So any of the living things. And when do we monitor? So monitoring can begin, for example, after a disturbance has occurred. So for example, after an earthquake or after a volcanic eruption and so on. And then the monitoring will be done to monitor the succession of plants and animals. But monitoring can also begin before a disturbance occurs, right? So for example, before an area is going to be cleared, before deforestation, before a construction site, things like that. So what can be done is scientists, they can monitor which organisms are living in the area and other information about the ecosystem. So they'll monitor it before the disturbance happens, and then they'll most likely monitor again afterwards. There are long-term monitoring programs as well. There's a species called an indicator species. So these are very important indicators of environmental change. So it could be a plant or it could be an animal species that helps to indicate environmental change. Amphibians like frogs, toads, and salamanders are very important indicator species because they are very sensitive to environmental change. If there's too much change, amphibians, they won't be able to survive. So a disappearance of amphibians would be a clue to scientists that there's something going on with the environment, which is not allowing the amphibians to survive. So things like pesticides, acid rain, loss of habitat, maybe an introduction of non-native species, they can all affect amphibians. And there is a decline in the numbers of amphibians and an increase in the number of amphibians that are being found with deformities. So that's a clue there that there is something going on in the environment. So monitoring an ecosystem over time, over many, many years, it gives a clearer picture of what is happening. Maybe there's too much pesticides being used in a farmland nearby. Too much pollution maybe is causing acid rain and so on. There's something called a baseline data. This is the information that's gathered by scientists to be used as a starting point to compare changes in the environment. So then that baseline data can then be used to predict changes in the environment. Permanent plots can be used. These are study areas that are used by scientists to monitor change. And then this is the report. It's called an environmental impact assessment. It's a report that will basically outline how an activity will affect the environment. Okay, we're just going to pause here and watch a few of these videos.
Have you ever heard the sound of frogs calling at night? For hundreds of millions of years, this croaking lullaby has filled the nighttime air. But recent studies suggest that these frogs are in danger of playing their final note. Over the past few decades, amphibian populations have been rapidly disappearing worldwide. Nearly 1/3 of the world's amphibian species are in danger of extinction, and over 100 species have already disappeared. But don't worry, there's still hope. Before we get into how to save the frogs, let's start by taking a look at why they're disappearing and why it's important to keep them around. Habitat destruction is the #1 problem for frog populations around the world. There are 7 billion humans on the planet, and we compete with frogs for habitat. We build cities, suburbs, and farms on top of frog habitat, and chop forests and drain the wetlands that serve as home for numerous amphibian populations. Climate change alters precipitation levels, drying up ponds, streams, and cloud forests. As the Earth's human population continues to grow, so will the threats amphibians face. There are a variety of other factors contributing to the frog's decline. Overharvesting for the pet and food trade results in millions of frogs being taken out of the wild each year. Invasive species such as non-native trout and crayfish eat native frogs. Humans are facilitating the spread of infectious diseases by shipping over 100 million amphibians around the world each year, for use as food, pets, bait, and in laboratories and zoos, with few regulations or quarantines. One of these diseases, chytridiomycosis, has driven stream-dwelling amphibian populations to extinction in Africa, Australia, Europe, and North, Central, and South America. On top of all these problems, we add hundreds of millions of kilograms of pesticides to our ecosystems each year, and these chemicals are easily absorbed through amphibians' permeable skin, causing immunosuppression or a weakened immune system and developmental deformities. Okay, so why are these little green guys worth keeping around? Frogs are important for a multitude of reasons. They're an integral part of the food web, eating flies, ticks, mosquitoes, and other disease vectors, thus protecting us against malaria, dengue fever, and other illnesses. Tadpoles keep waterways clean by feeding on algae, reducing the demand on our community's filtration systems and keeping our cost of water low. Frogs serve as a source of food for birds, fish, snakes, dragonflies, and even monkeys. When frogs disappear, the food web is disturbed, and other animals can disappear as well. Amphibians are also extremely important in human medicine. Over 10% of the Nobel Prizes in physiology and medicine have gone to researchers whose work depended on amphibians. Some of the antimicrobial peptides on frog skin can kill HIV, some act as painkillers, and others serve as natural mosquito repellents. Many discoveries await us if we can save the frogs, but when a frog species disappears, so does any promise it holds for improving human health. Fortunately, there are lots of ways you can help, and the best place to start is by improving your ecological footprint and day-to-day actions. The next time you listen to that nighttime lullaby, don't think of it as just another background noise. Hear it as a call for help, sung in perfect croaking harmony.
Even in the most extreme environments on Earth, there is life. These small, slow-growing living structures are called lichens. Lichens are often the first species to colonize an area. They forge their existence based on a partnership between two very different organisms: algae and fungi. The algal partner, capable of photosynthesis, manufactures energy for growth. The fungal partner has a large surface area to capture mineral nutrients. Its structure also protects the algae. Each pairing of a type of algae and fungi forms a different species of lichen. Lichens grow very slowly, as little as 1 millimeter a year, and may take up to 50 years to cover a square centimeter. They populate inhospitable habitats, such as bare rocks, barren deserts, and even the frozen polar regions. Lichens can survive in these hostile habitats as they draw water and minerals directly from the atmosphere over their entire surface. But this makes them extremely sensitive to atmospheric pollution. So there is much to be drawn from the study of lichens and where they are found. Pollutants in the air or dissolved in rainwater, such as sulfur dioxide, prevent lichen growth. So there are usually very few lichens around industrial centers and cities. This makes lichens natural indicators of air pollution. Lichens are considered among the oldest living organisms. They reflect the changing environmental conditions of our world. Perhaps most importantly, lichens show how organisms can thrive in harsh conditions through partnerships.
All right, so far we've looked at amphibians, and then we've looked at lichen species, how they are an important indicator species to indicate, for example, environmental change, pollution, and so on, which is affecting their survival and their growth. Our next video is another one on indicator species. I believe this one is on an animal species.
We're going to the top of the Colorado Rocky Mountains to visit with Dr. Chris Ray at the Niwot Ridge Long-Term Ecological Research Station. Chris Ray is studying the pika, an animal that may respond to environmental changes more dramatically than other measurements might recognize. This is called an indicator species.
I know these pikas being these fuzzy little creatures I see in the alpine when I go climbing. What are they?
Well, they're related to rabbits, about as big as your fist. Well, basically I study the effect of climate on the pika.
We're up here in the middle of winter when it's really cold. Why are we doing this? Aren't they fast asleep hibernating underground?
No. Pikas have to stay awake all winter. During the summer, they gather grasses and flowers and they stack it in a hay pile underneath some rocks. And they are burning those calories to stay warm right now. But they're also staying warm because they're covered with a blanket of snow. And what's great about studying them here is they have long-term data from these stations on wind speed, precipitation, temperature, even snow depth. Look at this snowfall! This thing is taller than me!
Wow.
And under all this snow, there's a hay pile.
So there's a hay pile down there. That means there's a pika down there.
That's right. It makes me wonder how they're doing up there.
So pikas live up in the rocks there too.
Absolutely, and up there's no snow cover. So how are they staying warm? The snow that you're standing on, it used to last sometimes into August, and now it's off and gone by May.
Wow.
And we think that's actually not so good for the survival of the pikas. The pika might be a potential indicator for the condition of the snowpack and the condition of the watershed in general.
Thank you so much.
Sure.
By observing pika populations, we may have an indicator for the health of our most precious resource, water.
Hi everyone, Ileana here. So for this next lesson, we'll be talking about indicator species and what they can tell us about a given environment. So indicator species give us clues about the overall health of an ecosystem. So down in the LA River, we have macroinvertebrates that live in the water. So a macroinvertebrate is an organism, so a little critter with no spine that is large enough, so it's macro, it's large enough for us to be able to see it without any fancy equipment. We could see it with our own naked eye. And there are many different types of macroinvertebrates down here in the water. And they give us clues about the environment, about the river, because there are many different macros, and some are tolerant, and some are sensitive, and some are in between. So when a critter is sensitive, that means that they can't really tolerate a lot of different conditions. They can't really tolerate when the water is poor quality. They don't survive, they can't make it, their shells may dissolve. And when a macro is tolerant, that means that they can handle different conditions, maybe conditions that are not so great. And there's also macros that are in the middle, so somewhat tolerant, somewhat sensitive. We could go down into the river and collect samples. We could look at the water and study what macros are found. And depending on what we find, we may find a lot of sensitive species, so that tells us that the water is good. Or we might find only tolerant species, so that may tell us that the water is not so great that day. And this is a very simple, inexpensive way to study the water quality, because we don't need fancy equipment really. All we need to do is go down there, maybe with a cup or a container. We could look for macros that are in the water, and they give us clues about a specific region of the river. And also macros don't really travel that far, so we could gain clues about an ecosystem. Okay, so when we go out to the river and we look for macros that live in the water, and we look for different critters, well, we are working as scientists. We are scientists out making observations. This is exactly what a scientist does. They first come up with a hypothesis, which is an educated guess, an informed guess, their best guess, and then they go out and they look for evidence. So evidence that will support their hypothesis and prove it right, or maybe prove it wrong, and they have to go back to their books and come up with a different hypothesis, and then once again come out to the environment that they are studying and look for more evidence. So it's a process, trial and error. So looking for macros is actually quite simple. All you need is a container or a cup, something to help you scoop up the water and get some samples. And you want to swish the water around. Macros like to hang out in murkier water, so you want to swish it around. And they also like to be around algae, because that's where they feed. So you want to swish the water around, you scoop it up, and you see what you find. And they're still very, very tiny. Even though you can see them with your naked eye, they are very small. So you have to look closely and see if you spot anything swimming around the water. I haven't found anything yet. Oh, I found something.
Oh, that's a big one.
Yeah. Because we're not so sure what kind of macro this is, we will be using our data sheets to help us identify. And according to our data sheets, we think that it might be a caddisfly larvae. So it is in the sensitive group. So it's a sensitive species, meaning that it is unable to tolerate poor conditions. So we are thinking that the water might be cleaner today, which is a good sign. It's a good sign when you find sensitive species. If you enjoyed these videos, please consider visiting our website.
All right, so that last video really got into how you can investigate conditions, for example, of a river using indicator species. So a species that's very sensitive to pollutants, it's not going to be able to survive. So because they were able to find a sensitive species living in that river area that day, they can basically say that that river condition was good quality water that day. But if they found species that are tolerant of pollutants and tolerant of certain conditions, then the water quality most likely would be poor quality water. All right, so the homework today is in the textbook. There is an investigation called What's the Change? And it's on page 71 in the textbook. So what's being done here is there's all this data on species in different years. So what you're going to be doing first is making a bar graph comparing the number of individuals observed in 1997 and in 1998. And then there's another table, table 1.2 there. Also, you're going to be making a bar graph just comparing the numbers of individuals that are found at 5 different sites in Alberta. So the 5 different sites are here. There's Beaver Hill Lake, Cypress Hills, Kananaskis, Lesser Slave Lake, and Meenok. So those are the 5 Alberta sites, and then there's different species being monitored. So LTSA, that's the long-toed salamander. TISA, that's the tiger salamander. BCFR, that's the boreal chorus frog. WOFR, that's the wood frog. CSFR, that's the Columbia spotted frog. NLFR is the northern leopard frog. BOTO, that's the boreal toad, and the last one, CATO, is the Canadian toad. All right, but when you make the graph, you just need to make the graph of the number of individuals that are found at the 5 different sites in Alberta. Once you do that, then there's this analyze. There's 3 questions there, just comparing, for example, what are the 2 most common amphibians in Alberta, which ones have the largest distribution through Alberta, which ones have the smallest distribution, and then the last question is just how can that data be used over long term. All right, so that is the homework for today.